Hello everyone, today we are going to talk about our skeletal muscles. We've got three types of muscles: skeletal, cardiac, and smooth. Cardiac and smooth muscles are known to be involuntary muscles. Skeletal muscles are voluntary muscles. When you look at the skeletal and the cardiac muscles under the microscope, you would notice that they have those striations, which we will go ahead and talk about today. Cardiac muscles have those intercalated discs that would help the muscle cells communicate pretty fast with one another and send electrical signals to each other through those intercalated discs. Quick review of the anatomy of our skeletal muscles. Skeletal muscles, as I said, are the ones attached to your skeletal bones. So if we have the skeletal muscle right here, this skeletal muscle is protected by a connective tissue membrane known as the epimysium. This muscle right here, it's made out of bundles. If you can look right here, there are those bundles. If you pull one of those bundles out, it's known as a fascicle. This fascicle right here, it's protected by a connective tissue membrane known as perimysium. Each fascicle is made out of smaller bundles. If you pull one of those bundles out, it's known as the muscle fiber, and the muscle fiber is actually our muscle cell. Muscle fibers are protected by a membrane known as endomysium, and if you actually remove the endomysium, there is another layer underneath it, and that would be the sarcolemma. Each muscle fiber, it's made out of a bunch of myofibrils. So here is one myofibril right there. There is no connective tissue covering the myofibril. You're going to have on top of the myofibril something known as the sarcoplasmic reticulum, and we will talk about it. Each myofibril, it's made out of a bunch of myofilaments. Those are going to be the protein filaments that are going to help our skeletal muscles contract and relax. And we've got two types of myofilaments. We have the thin filaments, or we refer to it as actin, and we have the thick filaments, or we refer to them as the myosin filaments. So this is a zoomed in image of a muscle fiber. As we said, our muscle fiber is our muscle cell. So protecting your muscle fiber, there is the connective tissue known as the endomysium. Underneath or deep to the endomysium, there is another layer known as the sarcolemma. The sarcolemma is kind of like the plasma membrane. So since your muscle fiber is a cell, each cell is surrounded by a plasma membrane, so we refer to it as the sarcolemma. The sarcolemma right here, surrounding the whole entire muscle fiber, parts of the sarcolemma kind of invaginates, as you can see, and goes deep, and we call those parts right here of the sarcolemma the T-tubules. So those light colored tubes right here, those are the T-tubules. The T-tubules are pretty important because when we start talking about how your muscles contract, we need to send an electrical signal or an action potential for those skeletal muscles to contract. And so those action potentials are actually going to make their way into the T-tubules. Next to the T-tubules, right on top of our myofibrils, we have this blue-like web sheath right here, and that is known as the sarcoplasmic reticulum, or SR for short. The sarcoplasmic reticulum is pretty important because this is where we are going to store our calcium ions, and calcium ions are pretty important for your muscles to contract. Specifically, the part that's going to store the calcium for you is right here. Those regions known as the terminal cisterns. Cistern meaning sac, and so terminal because they're kind of on the ends right here. So we usually call this region right here the triad because you have two terminal cisterns and in the middle the T-tubule, so they refer to it as the triad. Let's go ahead and zoom in on our myofibrils. So here is one myofibril. As we said, this myofibril, it's made out of a bunch of myofilaments. So all of these lines that you are seeing, those are the protein filaments that will help your muscles contract and relax. And as we said, we have two of them, the actin filaments and the myosin filaments. And this is why when you look at the skeletal muscles under the microscope, you see striations because you are actually looking at the myofilaments. Those myofilaments, they are highly organized and they create those block-like regions known as sarcomeres. So this one block right here is known as a sarcomere. If we zoom in on one sarcomere, right here you see the, let's look at those right here, those blue filaments. Notice how they're a bit thicker, so those are going to be your thick or myosin filaments. And then those red lines are representing our actin or our thin filaments. Notice how they are highly organized, one myosin on top of an actin, or you can think of it as the actin sandwiched in between two myosins. Now, those actin filaments, they are attached to proteins known as the Z disc. From one Z disc to another Z disc, this is one sarcomere right there. This region right here, where you only have thin filaments, we refer to it as the I band. This whole entire area where you have both filaments or all of your filaments, both myosin and actin, we refer to it as the A band. In the middle of the A band, there is, you look right here, there are only myosin filaments, so we refer to this region right here the H zone. You can remember H zone only carrying heavy filaments or the myosin filaments. Um, when a muscle is relaxed, you can see the H zone. And in the middle of the H zone, by the way, there is this line right here, which we refer to it as the M line. It's just another protein grabbing onto the myosin filaments. So when a muscle is relaxed, you can see the H zone and you see that the actin filaments are not reaching the M line. When you start contracting your muscle, we go through something known as the sliding filament theory. And why is it called sliding? Because check it out, the actin filaments are actually going to start sliding towards the M line or towards that H zone. So that's why we call it the sliding theory. So right here, this is an image of a relaxed muscle. You can see the H zone right there. Or if you look at this histology image, here is one Z disc and here's another Z disc. So this is one sarcomere right there with the H zone in the middle, and you can see the I band right there. When a muscle starts to contract, and you have full contraction, you notice that your I bands are going to actually get smaller. And why are your I bands getting shorter? Because your I bands are attached to the Z disc. So when the Z disc basically gets pulled closer, so the Z disc is basically kind of moving towards the M line. So when the Z discs, they move closer to each other and they are both heading towards the middle of the sarcomere, the M line. Of course, the I bands on the sides are going to get shorter. Now, nothing happens to the size of the actin filaments or the myosin filaments. They stay the same size. All what's happening is that the actin filaments are just sliding towards the middle of the sarcomere. That's all what's happening. But so nothing is going to happen even to the size of your A band. The A band stays the same size. The H zone is going to disappear. So the H zone in the middle, as you can see right here, you're not going to have any more H zone because now you have full overlapping between the actin and the myosin filaments. This is just another image showing you your Z discs right there. So here are the Z discs, and you can see a pretty large I band. Again, those are the actin filaments right here. Now, when a muscle starts to contract, those Z discs are going to start moving inwards. Why? Because the actin filaments are being pulled inwards. So as the actin filaments are being pulled towards the middle, the Z disc, of course, is going to follow along since it's attached to it. So you can see right here the difference. The Z discs get closer and the I bands are getting shorter or smaller in size. And no more H zone because you can see that the actin filaments slid all the way towards the M line. And that's what's known as the sliding filament theory. Now, who's pulling the actin filaments towards the M line? The myosin filaments. So as you can see, when a muscle is relaxed, there is no contact between the myosin and the actin. But once contraction starts, the myosin filaments are going to grab on to the actin filaments and start pulling the actin filaments towards the middle. So right here, this is a more zoomed in image on our myosin and actin filaments. And you can see right here, there is no contact between the two, so your muscle is relaxed. If we zoom in on our myosin filaments, this is how they look like. There is a tail and there are heads. So we've got like two heads and two tails, and they're kind of like twisted around each other. Those heads right here have two binding sites. Right here, ATP is going to bind to the myosin head, and we're going to discuss in a few why it's important for ATP to bind. And then right here, there's a binding site for actin to grab on. So right here, actin is going to bind, and on the side right here, ATP can bind. If we look at the thin filaments, we have two strands. You see right here, those two blue strands, we call them F-actin. And we can see that each F-actin strand, it's made up of a bunch of beads. You can think of them analogy-wise. If this is a necklace, then those right here are the beads. So the beads are what we refer to as the actin subunits, or sometimes they refer to it as the G, the letter G subunit. Each subunit, each actin subunit has an active site. You see this dent right here? This is where the myosin head is going to grab on. Once myosin grabs on to those active sites, your muscles are going to start to contract. So if you think about it, do we want those active sites to be exposed at all times? If they are exposed at all times, then your myosin heads are going to grab on all the time, and your muscles are going to be in a contracted state all the time, which is tiring. So to allow your muscles to relax, you don't want those active sites on your G subunits to be exposed all the time. And so another protein that's pretty important to talk about, known as tropomyosin, is responsible for covering up those active sites on your G subunits. And as long as those active sites, as you can see, are blocked right here by the tropomyosin, then your muscles are not going to contract. For your muscles to contract, tropomyosin has to unravel to expose those active sites. Well, the question is, when is tropomyosin going to unravel to allow your muscles to contract? And the answer is when calcium ion, binds to another protein right here within your actin filaments known as troponin. Notice how troponin is bound to tropomyosin. So when calcium binds to troponin, troponin is going to change shape. When troponin changes its shape and it's attached to tropomyosin, it's going to force the tropomyosin to move away, to unravel, exposing the active sites, and the muscles are going to start to contract.